Okay, so yeah, feel free to interrupt me during my talk. Yeah. So first, I want to say that Vivian, thank you so much for organizing this great seminar with a kind invitation. I think that was right after our preprint on the bioarchive. Anyway, yeah, let's get started. So yeah, today's my talk. Before that, yeah, just. Oh, slide doesn't work. Okay, so now I'm in the NYU Langone, in Manhattan. So the NYU Langone is located around the East River, and there is a couple of buildings because this is a big medical center. So also I am in the Diolin's lab. So she is focusing on the social behaviors circuitry. So I think that she is around here, I guess. Yeah. The 13th floor of this building. So today I want to share with you our latest preprint, oxytocin signaling as for the social aversive behaviors. So, okay, let's get started. So in animal kingdom, as you know, that there are the kinds of social interaction among species, not only human social gathering, but also interaction between parents and kids, for example, the top right, the penguins showing like that. And also there are the interaction, a lovely interaction in dogs or the mice or the monkeys here. But anyhow, the social interaction is essential for us to bring our daily life smoothly. So I also want to show one example in human. So here is the two boys, and initially boy approaches towards the other and say hi or something and has communication. However, if he or she is a bully, what's going on? So on the next day, the bullied boy will show avoidance from the bully when he finds the previous friends or something. So anyway, we could easy to imagine the plasticity of social interaction and the drastic behavior change in humans as well. So today's talk is in mice. So I want to show two videos here. Okay, so how about, sorry. Yeah, this assay is, cupped aggressor. White mice in this study is aggressor, and the black mice is test animals. So this left video is the before a social defeat. So black weaker, honestly weaker than the aggressor, but black test mice easy to show approaching behavior towards cup, and there is the sniffing behaviors around the cup. So the second video is also the one cup assay, but after defeat behaviors. So after defeat experience, initially the black mice attempt to approach, but showing the leechy behaviors and staying longer duration on the other side of the cup. So as I told you, the bully or the bullied children in the before, but in mice there is also the same situation. We can easy to observe. So this is just a heat map of their behavior. Let's see. So the question, today's question we want to address is which circuitry is responsible for this social avoidance behavior, this plasticity in social behavior. So social avoidance is in animals, experience a defeat. So responsible circuits for avoidance behavior could be highly activated during defeat as well. So there is some brain lesions activated during defeat events, but here I want to introduce one of the lesions in the hypothalamus because we love have a special eye on it. So this lesion is, shown on the top, the ventral pole of the ventral medial hypothalamus. So BMHBL, we say that BMHBL is a key brain lesion for social behaviors. It controls the aggressive behaviors in both male and female. and also this lesion control sexual behavior as well. In addition to those behaviors, the defeat is also controlled by the BMHBL. So this data was in previous studies. These data are represented to sections with CFOS immediate origin, staining after social behaviors. And this images from anterior, left side is anterior and the right side is posterior section of the BMHBL. So after defeat, you can see that the dense cluster of the white dense cluster of the activated cells in the anterior, especially anterior part of the BMHBL. While after aggression, I just added, is CFOS after aggression is evenly distributed in the BMHBL. So from our previous data, we can say that the anterior part of the BMHBL could be potentially for the defeat events. So then the next question should be the relevant molecular marker for anterior part of the BMHBL. So there's many candidates, but today we want to focus in on the oxytocin related molecules. So this image is a section from oxytocin receptor animals. So closed with the AI6 reporter line. So the oxytocin receptor now here is showing green. So anterior part of the BMHBL is ventral part of this section. So there is allos on the bottom. So anterior BMHBL, we can see dense cluster of the oxytocin receptors. So in this study, we wanted to focus in on the importance of the oxytocin receptor. Later on, I want to show the oxytocin signaling. Oxytocin secretion itself is also important for social avoidance behaviors. So here I want to show results of the staining in the BMHBL. So there are the green cells representing oxytocin receptors with reporter mice, and the red cells representing CFOS after social behavior. So red left side is attack, and the right side blue is the after defeat. So we can find that there are many CFOS induced cells in the anterior part of the BMHBL. Anterior part of the BMHBL showing on the top after defeat. So these data also supported by the previous study focusing on defeat related circuitries. So next we performed was the recording of neural activity with fiber photometry doing social behaviors. Here we used the left side, there was the scheme. We used the oxytocin receptor Curi animals with GCAM virus, GCAM expressing virus and optic fibers on it. So this first video is the black test mice interact with weaker, even though white, but this intruder is weaker animals, Barbary male. So test black mice initially showing approaching behavior, and this encounter is weak, so there should be the biting events. So yeah, right now there is attack, but even though there is some biting events, but sorry, I have to say in advance, but on the bottom, there is the chart of the neural activity of the anterior part of the BMHBL oxytocin receptor population. So when the animal interact with weakers and with biting, but there is no neural activities in this region. So let's show the next video. Next video is with white but the stronger aggressor Swiss strain. So okay. So initially, this is his home cage. So initially, black mice, test mice approach towards white, but white SW strain is quite strong. So there was events with being bitten. So as you can see, there is strong neural activation when he was bitten. So this is quantification of the videos, GCAMP recording from the oxytocin receptor cells in the anterior part of the BMHBL. So the left one is the interaction with weaker Barbusi male, weaker male, and during attack there is almost nothing. However, with a stronger male and with defeat events, there is the strong neural activation when he was defeated. So, okay. So we also try other types of behaviors. So we still use the GCAMP animals, but this test is the test with the bigger arena. So in this arena, there are 4 cups on each corner. So the one with the weak male, and there is the same strain and the stronger aggressor SW, and this is just an empty cup. So, okay, let's get started with the before defeat. So the animal moves freely in the arena and interact with each cup. Also, during this his visits, neural activity of OXTR is still silent. Okay, so next one is 1 day after defeat. Sorry. Okay. So in contrast, so this videos after defeat, empty cup and same strain and SW. So there is quick retreat with the strong increase of neural activity. So this data is also suggesting that there is the strain specific neural activity after defeat events. Okay, so now let's check the result from this bigger chamber assay. So left side, bottom of the left side is the heat map of the body tracking, says that the visit around the aggressor cup, bottom right here, seems to be decreased. And this is also quantified by the human notation of the total time around the cups showing on the right side. So towards the other cup, there is the same time visiting, but visiting towards the aggressor seems to be decreased. Okay, so this is the data results of the neural activity from the big arena assay. So there is no change when animals visited the cup of empty or Barbusi weak male or the same C57 male. However, there is the significant increase after defeat during the visits towards the aggressors. So these data, both data show that there is a dynamic change of neural activity in anterior part of the BMHBL oxytocin receptor positive population after defeat, specifically towards the aggressor cues. So next, we want to address was this drastic change in with the in vitro stuff using the slice EFIS. So Longgen postdoc in our lab performed the recordings. So here we use the oxytocin receptor reporter mice and made the 2 conditions for the recording. SH here means just a single house animals, and the bottom D means the animals with defeated events. So here is the data. So when we perform the recordings from the receptor, oxytocin receptor positive cells, same in the anterior part of the BMHBL, we found that the increase of spontaneous EPSC. And this data supports that the GCAMP, this data supporting that the GCAMP recording I previously shown to identify the change of their properties after defeat events. So, okay. So far was the neural activity and the characters of the oxytocin receptor positive population in the BMHBL. So from now, I want to talk about the neural activity itself of oxytocin receptor positive population in the same BMHBL anterior part. So first is, what is the increased BMHBL, anterior BMHBL cell response after defeat is good for. So to address this question, we express the CHR2 channel rhodopsin in the same region. So here's the video. So first half, there is no light, so he can approach towards the cup. But once the light has turned on, he shows a retreat, even though there's approaching behaviors, but he showed the quick retreat during the light, turning on the light. It means the activation of the neural activity. Okay, so let's show the quantification. So when we do two groups, the GFP control and the CHR2 activated groups, so the green here is representing the neural activation of the oxytocin receptor positive clusters. So there is the decrease of investigation during the right on, and there is also increase of the immobility when we activate this population oxytocin receptors in the BMHBL. So this was the neural, activation of the neural activity in the BMHBL. And next is, loss of function. So there is kinds of method I want to show. First is the inhibitory opsin. So yeah, inhibitory anion channel rhodopsin. So here is the ZTACR number 2. So we express the ZTACR bilaterally and shine the light through fibers. So the scheme here is the light, shining the light one day after defeat in order to, check the behavior change after defeat. So first video is just control. So control animal, even though there is a light, but there is strong avoidance behavior because the virus is just arbitrary. So in contrast, this is test group with inhibition. Okay. So initially they approach and he's still staying around the cup. Usually the animals easy to show quick retreat and avoidance behavior, but when do inhibition of this population, there is long sleeping. So there is less avoidance behaviors with inhibition. So this is just a quantification, but inhibition of this population make less avoiding situation. So we also check the same thing with the dread. So here we also inject bilaterally inhibitory dread virus here. So, okay. So this is the tracking, nose tracking from the control group. So the top is eventually control virus with. So here we used was agonist 21, the artificial agonist of the dread. So in any case, the M-cherry with agonist 21 or the GI virus with saline, just a vehicle, both groups showing strong avoidance behavior. So the dots, the nose tracking around the cup is significantly decreased. So the third one is the inhibiting group. So GI dread with agonist 21. So in this group, we can see that the dense clusters nose tracking, even though after defeat. So this is quantification, but this inhibitory dread data also supporting that the when inhibiting this population, there is less avoiding behavior towards aggressors, even though they experience the defeat by strongers. So, okay, so here I want to show our working model. So, okay, so before defeat, even though there's the aggressor cues, but the neural activity of anterior part of the BMHBL is quiet. So there is no specific behavior output. In this case, they show approaching, but no avoidance behaviors. After defeat, the mice would show prompt avoidance behaviors when they re-encounter the aggressors, because the anterior part of the BMHBL oxytocin receptor positive population are now strongly activated because of defeat experience. So the next question is what's going on during defeat? During defeat, there should be the multiple cues, for example, the aggressor odor from the aggressors or the pain signal when he or she was being bitten. So here, the one we wanted to address was whether oxytocin receptor itself is important to avoidance behaviors learning or not. So to check whether oxytocin receptor itself is important to induce social avoidance, we use the genetical knockout to make lesion-specific knockout. So those animals are after defeat, so left one is control. So yeah, so once he's get some odors from the cups, there is strong avoidance because of this animal is just a control. So how about the knockout in the anterior part of the BMHBL? So approach and sniffing, and there is some, but in this video, it's difficult to see the strong avoidance behaviors. Yes, still sniffing, even though he experienced the defeat one day before. So, okay, yeah, I want to show here is quantification of this lesion-specific knockout. So, cup assay was performed before and after defeat events, and we compare the percentage of approaching behavior towards the cup. So here, the control group showing gray only show the 20% of the visits towards the aggressor's cup. But however, the lesion-specific knockout, there is not the 100%, but the avoidance behaviors are going to be much more weaker than the control. So that means that this oxytocin receptors in the anterior part of the BMHBL is important for showing avoidance behaviors. And this was also same analysis by using the female mice. In female case, we use the mothers as aggressors, and the same trend was observed. So this trend, it means importance of the oxytocin receptors in the anterior BMHBL is common between male and also females. Okay, so the previous one with lesion-specific knockout suggests the importance of oxytocin receptors, and then we have done the assay with application of agonist through the implanted cannula onto the BMHBL. So here's the paradigm. So the drug in this slide, the drug infusion is 20 minutes before resident intruder assay, before 20 minutes before defeat. So it means that during defeat, the function of the oxytocin receptor was antagonized. So let's see. So when we check the data, the antagonist applied animals, when we apply the antagonist, the approaching behavior towards the cup has increased. That means there is less avoidance behavior in the group with antagonist. So this seems to be the same paradigm, but when we apply the antagonist is a bit different. So in this slide, we applied the drug 20 minutes. Sorry, this 20 minutes is place of 20 minutes is weird, but sorry, the 20 minutes should be here. So 20 minutes before the cup assay. So we want to antagonize the receptors during fear expression period. So here is the data, but even though we inhibited the oxytocin receptors during the fear expression, but both group control and the receptor antagonist group show strong avoidance behavior. So that means the oxytocin receptor itself is important to show fear acquisition, but for the fear expression, oxytocin receptor is not important during this period. So yeah, from our second part, before defeat, it's fine. I told you before, before defeat, there is no avoidance behavior because until BMHBL is very quiet. And after defeat, there is aggressor cues because of encountering with aggressors and strong prompt avoidance behavior. So let's check during defeat. So there should be the pain signal and the aggressor cues. But now we have shown here was the oxytocin receptor itself is quite important to acquire the information and important for the to show the prompt avoidance behaviors. Okay. So the last question I want to show today is whether oxytocin peptide itself is important to show avoidance behavior or not. Okay, so when we want to consider the oxytocin secretion, there should be some potential region can secrete the oxytocin peptide towards other region. So there is 3 region here, PBN. I think PBN is a very popular region with oxytocin expressing cells showing that. And the SON, supraoptic, there is tiny region aside of the anterior part of the medial amygdala. And also there is some private lesion, the retrochiasmatic supraoptic lesion. So we wanted to target these 3 regions for oxytocin expressing population. So now I added was the CFOS expression after defeat events. So on the right side, there is the percentage of overlap between oxytocin positive cells and CFOS neuroactivation after defeat. Of course, there is a lot of overlap, but when we compare the percentage of overlap, this very tiny region, but the SOR show higher overlap other than 2 regions, PBN or SON. But still there is some overlap in PBN. So next we wanted to address was with EFIS. But before that, yeah, I want to say that this SOR region, a very tiny region, is also interesting from its location, anatomical reasons. So the SOR here showing red is very close to anterior part of the BMHBL. First part I talked about anterior part of the BMHBL, and now the oxytocin receptor is shown in green here. So from this anatomical situation, it is very interesting to see relationships between oxytocin receptors in the anteroparietal BMHBL and the SOR. There is the oxytocin expressing cells. So now we wanted to test directly whether these cells are important for oxytocin neuroactivity of oxytocin receptors in the anteroparietal BMHBL. So we, in order to perform check the things, we optogenetically activate the SOR oxytocin expressing cells. So these animals is oxytocin mice and the Cre-dependent channelrhodopsin expressing virus. So SOR oxytocin cells are labeled by the CHR2, and the patch was onto anteroparietal BMHBL cells. So yeah, now we can see that when we activate SOR clusters, oxytocin expressing population, there is, even though slow, but the depolarization of the BMHBL population. So, okay, so next. So how about the PBN? Because from the staining data, there is some overlap. So we wanted to address this lesion as well to confirm whether PBN is still important or not. So we perform the same paradigm. The virus expression CHR2 virus was PBN, and patch the cell from the anteroparietal BMHBL. So when we shine the light, but there was no depolarization, even though there is the optogenetic neuroactivation with blue light. So yeah, those two data suggesting, oh, here is the quantification, but those two data suggesting very timely lesion, but the SOR is a potential fly-based lesion for the oxytocin receptors in anteroparietal BMHBL, but the PBN is not for supply. So next we wanted to address here was the role of the release of the oxytocin in the anteroparietal BMHBL. So here was a little bit tricky paradigm, but we used the virus-mediated paradigm to see whether there is the LTP induction or not after oxytocin, private oxytocin supply. So PA, posterior amygdala here was, we already know that the PA sending the excitatory axon towards the BMHBL. So in this model, we use the PA as upstream, mimicking like, the detecting the information of aggressors. So those information potentially are sending sent it from the PA towards the BMHBL. And green cells is just a BMHBL population with the portal line. So anyhow, we wanted to see the plasticity of this synaptic connection from PA towards the BMHBL. Yeah, so yeah, with or without TIGO application, we want to check the situation. TIGO is oxytocin, oxytocin, not the receptor, oxytocin agonist. So, we want to see the difference between with or without OXT effect. So here is the data. So without any oxytocin agonist, there is no change of the EPSP. However, when we apply the TIGO oxytocin agonist, there is strong significant increase of the EPSP. So, here is the quantification, but with application of TIGO, there is the increase of the EPSP with the firing probability suggesting that the oxytocin release at the around the BMHBL facilitates the potentiation, synaptic potentiation. So, this paradigm mimicking the when they animals be encounter the aggressors, there should be the mechanism of the LTP working for prompt avoidance behaviors. So, okay, last part is, neural recording and the functional stuff onto SOR, oxytocin expressing population, oxytocin releasing population. So here is the GCamp onto SOR. So, using the oxytocin cleave with GCamp expressing virus. So, okay, let's check the first video. So with a weak intruder. So I think easy to imagine, but there is black mice showing biting events towards weakers, but this population SOR OXT is quiet. Okay, let's go to second one. So this white, but the SW aggressors. So initially, sorry, this is not the first defeat event. So there is kinds of avoidance behaviors, but when he was beaten, there is strong induction. Overall, this population, SOR or XT cell, is quite, very quiet. But when he was defeated, there is striking activation. Okay, so this is just a quantification from the GCamp recording. So with weaker intruder, there is no induction, even though there is aggression. So the right side is interaction with stronger SW male. So with defeat events, there is induction of neural activation aligned to defeat events. So the next question is, what's going on when they re-encounter the aggressors? So the question is SOR OXT population could be activated by the cues from aggressors or not. So here is also the big arena assay for cup empty. The position of the distribution of the cup is a little bit different, but so this video left side is before defeat. So animal easy to approach, even though in the cup there is aggressors, but and this population is very quiet. Okay, so the next one is 1 day after defeat. So empty cup. Yeah, so now he showed the strong avoidance behavior, quick retreat when he touched, crossed by the strongest cup, but the neural activity of this population, SOR OXT, is quiet. So this population is important during defeat events, but after defeat, when they showing the avoidance behavior, this population is is not activated. So when showing the avoidance behavior, as I shown before, the LTP effect is void, but the SOR oxytocin release is not anymore doing avoidance behavior itself. Okay, so the next. So here we wanted to address was, we observed the neural activation during defeat events. So which kind of factors during defeat or being beaten oxytocin expressing population in the SOR detecting? So with the urine from aggressors or just gentle touch, there is no activation. But when we did the back pinch or the poking or the tail pinch, there is strong neural activation in this tiny lesion SOR OXT population. So this is just a quantification of C-score. So, and the same trend was also observed in female animals. So as I shown before, the phenotype of oxytocin receptor, importance of oxytocin receptor is common between sexes. And here we also wanted to say that the function of SOR OXT population is also common between both sexes, male and female. Okay, so let's go to next. So next two slides is functional studies of SOR population. Here we wanted to perform was the cell ablation with diphtheria toxin mediated strategy. So when we inject the virus and when with drug application, the oxytocin itself is aberrated. It's in SOR, there is the oxytocin population and also the vasopressin population, but this gene-specific aberration working for oxytocin cell. So, and we also perform the behavior stuff, the GFP, just a control, and the ablated group. So DTL showing green, ablated group showing less avoidance behavior. So there is still, the situation is not the 0 or the 100% or nothing, but yeah, we can easy to imagine that the central amygdala or the other region is also for the avoidance behavior. There should be the parallel circuits, but we want to say here is SOR and the oxytocin receptor circuitry is important for perfect or the very strong avoidance behavior. So at the end, we performed was the gain of function studies onto oxytocin expressing cells in the SOR population. So we expressed the CHR2 bilaterally and optic fiber was placed above the lesion. So in this paradigm, there is some trick because we want to see the induction of the increase of the avoidance behavior. So there is the brief defeat events. So only 3 or 4 times defeat episode with light with light stimulation to make the higher baseline. So when we do this assay with control animals, just the GFP injected animals, the both two groups showing less avoidance behaviors. But when we shine the light during the 3 or 4 times defeat events, there is the increase of the avoidance behavior. So in this case, the baseline is around 80%. That means almost no avoidance behavior in control group or the light off. But when we shine the light onto the experimental group, the baseline 80% going down to 40, that means the induction, neuroactivation of SOR population could promote the avoidance behaviors towards the aggressor cup. So now I want to wrap up with summary. So yeah, this is third time, so easy to imagine, but before defeat the aggressor cube, but there is no avoidance behavior until part of the BMHBL population. Oxytocin receptor positive population is originally very quiet towards the aggressor cube. During defeat, there is the aggressor cues and also the pain signal, and the pain signal at least partially onto small clusters, SOR, oxytocin positive population. And there should be the oxytocin peptide release, and the peptide itself should be onto oxytocin receptor itself in the anterior part of the BMHBL. So, and we check with if there should be the synaptic potentiation onto anterior part of the BMHBL OXTR population. So then after defeat, when the animal re-encounter with aggressors, the aggressor cues EC to activate this same population, even though same, but the LTP can induce the neural activity of this anterior part of the BMHBL population or XTR population. And then the animal is to show prompt quick avoidance behavior to avoid further difficulties. So yeah, this is the summary of our studies. Now is in Philippines, but yeah, I want to acknowledge the research funding. So this project is with the uniting program among the NYU Langone laboratories, so such as the Yuli or the Loeb or the Dick Chen. So yeah, so these big funding supporting these studies, and initially there was question why oxytocin is originally there is couple studies is running because of this funding. And also I want to say thank you for my private funding from Japanese organization. And yeah, and I also want to say, members in the uniting group, the image pictures is with the members of the Lynn's lab, a little bit, not the latest one though, but especially the Longgen, the postdoc working for the APHIS, and the Ewing lab manager right now is working on the female analysis. And yeah, also there's many inputs and help from other members in not only in the Lynn's lab, also from the uniting collaborators. So yeah, I want to say thank you for all of the members, not only for the authors, but all members for accomplishing these studies. So anyway, yeah, this is the end of my talks. Thank you for listening. So let me know any questions, right now or later.
So now it's the question time, and if you would like to ask a question yourself, please just unmute, or you can just put in the chat. And we already have Vivian, who is also one of the best speakers, and then you can find her talk on our YouTube channel. Vivian, please ask your question.
Great, thank you so much. It was really interesting.
Thank you.
My student had a question first.
Yes, yeah. So I was wondering first if you could tell me how old the mice were, and if you think that maybe age also would show difference in the response in behavior.
Sorry, the second question is depending on the age, you mean?
Yes.
Sorry for, could you say the first one?
The first one, just simple. At what age did you test?
I see. Got it. So yeah, honestly, depending on, but basically we use let's say the 9 weeks to 12 weeks for the white type assay, but there was the manipulating assay as well, is right? So in that case, we inject the virus or the cannula around 8 or 9 weeks. Then we have to wait 3 or 4 more weeks for the viral expression. So let's say behavior from the white type is around 8 to 9 or 8 to 10, but our manipulating stuff is a little bit older than, let's say 12 to 14 weeks or something. But yeah, anyway, yeah.
All right. And also, just I don't know if I got this right, but are they scared of any mice that they think is more aggressive than them, or is it a specific mice that how does it work?
Yeah, that's question. The thing, you know, your idea is same strain, but the exact aggressor or not is right? No.
Yeah.
Yeah, that's good question. Honestly, now we are addressing that question. My impression so far is potentially they can, but you know, I think the odors from the aggressor, now we use the SW strain, Swiss strain as aggressor. I think just my impression, but the odor cues from the you know, exact the XW is a little bit stronger than others. So my impression right now, you know, we are working with CUP stuff to check, you know, discrimination between animals. There seems to be some, but not, you know, striking because of the, let's say, common odors from, you know, all SW males or females seems to be masking, you know, individual, you know, difference. So probably I think, you know, there should be more, you know, dedicated way to check it. So our behavior paradigm is just cup, that's it. So if we establish more precise way to address, I think, yeah, there is more, you know, striking the difference. But my impression is there is some, and we can show some difference between. Anyway, yeah, that's a good point, I guess. Yeah.
Okay, cool. And then I think last question.
Thank you.
Yeah, I was just I was just wondering, so if they were or when they were attacked, and they are more scared after this plasticity, how long does it, do you know how long that lasts?
Yeah, that's also, yeah, important question. So first, I have to say is it depending on the situation, you know, how many times they were defeated, or, you know, how strong it was. So in our studies, basically we use the paradigm of 10 minutes. That means around the 5 second, in total, 5 second, you know, biting events. Biting means, you know, just a, you know, interaction, you know, be between by strongest is around 5 seconds. In that, also the other factors I have to mention is it depending on the animal itself, because some is very receptive, but some is resilient towards stimuli, right? So, but basically, I think this, you know, effect is long lasting with 3, 4, 5. So it depending on the animals, because some of them is, you know, easy to approach, even though there's tons of defeats. So there is variability between animals, but yeah, usually I think 3, a little bit shorter, yeah, 4 weeks or 5 weeks is needed for, you know, fully recovered.
Okay, thank you so much. It was really
Thank you so
interesting.
much for listening and questions. Thank you so much.
And we have another question in the chat.
Okay.
Rosa wanted to know if the oxytocin neurons in the PVN also regulate this submissive avoidance behavior.
So, okay. Yeah, so this is also a good point. So from our EFIS data, we want to say no. So because the, you know, importance of PBN is not for this avoidance behaviors, but yeah, potentially there is some clusters can be activated by defeat itself. So because, you know, PBN OXT population is heterogeneous, so we just performed was the some, no, I haven't shown here, but some GCamp and EFIS. From the EFIS data, I think there is no strong effects, but if we do some single cell level analysis, yeah, there is some, but I think SOL is one crucial region for, yeah, onto PMHBL.
Okay, and I have one last question.
Yeah.
In Fly, when the animals are beaten, there's a effect called the loser effect. You will continuously lose the next battle or have a chance to lose the next battle. Just keep losing by the end of it. I was just wondering whether, I know that in your paradigm, you have only used one shot of defeat as a way to create this social avoidance behavior. So I was just wondering whether you have sort of planning to test whether there's any subsequent behavioral change or long-term behavioral adaptation by the losers.
Yeah, so that's a good question. So in my case, honestly, first paradigm was the 3 days consecutive defeats. That was quite stronger. For example, G camp is very stable and the behavior is quite strong. But for the honestly, there is some practical reason. But if with 3 days stuff, manipulation is sometimes a little bit difficult because baseline is quite low, is right. So we finally use the 1 day defeat paradigm to show the importance of the receptors or oxytocin cell. But anyway, as for your question, 3 days defeat, for example, is much more stronger behavior effect. And even though there is stronger, activity in the, at least in the BMHB oxytocin receptors. So yeah, we just try was 1 day, 3 days, but there is some positive correlation. Yeah, I guess.
Yeah, thank you. I think we have more questions from Vivian.
Thank you. Sorry.
No problem.
Yeah, so I was also wondering, what exactly is the mechanism that comes from the nociceptors and go to the sore area? Like, where does it connect?
So you mean the downstream?
Yes.
That's good point. So honestly, we've just tried was response of SORs, right, for the noxious stimulation.
Yes.
So yeah, that's good point. So noxious stimuli, first noxious stimuli is not only onto SORs, right? Some sorry, I forgot the exact name. Some posterior region also detecting the pain signal itself and sending the information towards the BMH dorsal region of BMHD or something. So in this case, we just shown here was the SOR detecting the pain signal and that potentially releasing the oxytocin peptide and information onto BMHBL. So our paradigm today is SOR oxytocin expressing cells towards the receptors in the BMHBs, right? So yeah, the other thing, SOR population, we also check the projection pattern of the SOR, but quite limited. So I think this is very famous, but the SOR or the PBN sending very strong axon towards the medial eminence, or let's say pituitary is right. But brain, in the brain, there is some axons around the BMHBL, for example, or the, let's say, but in any case, I want to say it's quite limited. So in this case, our paradigm is SOR detecting and the oxytocin release from there and the anterior part of the BMHBL. That is, just a one lesion for detection, but there should be other regions also for pain sensation. So there should be some interaction.
Yeah, I was wondering if it was like a kind of like a more direct nociceptor signaling, or if it was through like sympathetic activation after the pain or something like that. But
so I want to say oxytocin release itself is only from the SOR, then receptors. But yeah, I'm not quite sure about interaction, but that is very interesting because, finally BMHD, BMHBL, as I shown here, information somewhat from the posterior brain region towards BMHD and the SOR towards, BMHBL with oxytocin release as well. So there should be some, interaction or integration of noxious or the pain signaling. So that should be very interesting, and potentially BMHD or BMHBL is working for it. So now it's just a purse of information, but yeah, let's see.
And just one last one, I was wondering, like, so I guess the whole system is made so that when we meet something that is dangerous, we like avoid it, and even more if the interaction was painful. So I was wondering, are there other signals, like, instead of like potentiating this area, are there areas, if we actually realize that this interaction was not painful, there was no problem, like, are there other areas of the brain that quenches down the sore, for instance, and kind of decrease this interaction between the amygdala and the hypothalamus?
So the question is the effect from other regions. Sorry, could you?
Yeah, because in this context, the sore area is potentiating the hypothalamus to receive inputs from like the amygdala, for instance. I was wondering, are there mechanisms to decrease the sensitivity?
Decrease the sensitivity of, let's say, the aggressor cues, you mean?
To decrease the potentiation, decrease the sensitivity of the hypothalamus to the amygdala input, like the opposite.
Yeah, so let's say, yeah, I see, that's good. Honestly, our idea is not decreasing the, your idea is potentially BMH hypothalamus towards amygdala region, potentially decrease their effect is right. Let's say, or the other.
So if I understood correctly, in this context of pain, the signal from the amygdala to the hypothalamus is potentiated. There's more CFOS activation, there's more excitatory signal in the hypothalamus, right?
I see. So
I was wondering if the opposite could also happen in case that there's no pain signal, there's, I don't know, happy signal or satiating or dopamine or I don't know.
I see. That's a good point. So at first, yeah, for the cues from aggressors, hypothesis is, the cues, odor cues from, let's say, the amygdala region, but especially posterior amygdala or in the hypothalamus, but the PMB should be the upstream. So let's say PMB towards BMHBL or the PA towards the BMHBL should be the projection or the, information signaling as for odor cues. So usually, these circuits detecting and discriminate or integrate the information, and there is no reciprocal connectivity. So sorry, just potentially there is, but yeah, but the other way we haven't dig into, so it's good to see. And the second, so first part is, our hypothesis idea is the odor cues should be from PA, posterior amygdala or PMB, and those information has changed with LTP in this case or other mechanism. And the other way, reciprocal connectivity stuff, it should be checked, but yeah, us or the other group haven't checked precisely, so it's good to check the other way. And the second part is that even though amygdala, but there should be the central amygdala or some things, right? So the BMHBL towards central amygdala, there should be some projection as well. So as for the fear learning or fear expression, avoidance expression, I think those those projection, BMHBL towards, let's say, CEA, for example, is potentially important For example, for freezing, honestly, we have to check a little bit for it because of the revision. But even though amygdala, but for the inputs or the some freezing behaviors, there is some distinct routes, I guess. But yeah, as for your question, we haven't. But yeah, there can be some ideas.
Thank you. Thanks a lot. It was really
No problem. Thank you. Yeah, whenever, please, ask me. Later on. Ask me.
Cool. And so because of time, and we just wanted to thank you again for being here, for wonderful talk and wonderful questions. And then we can definitely stay on if you wanted to continue for discussion, but just let's give Takuya another round of applause and thank him for a wonderful presentation. Thank you.